Good morning to each one of you present here today. It is a moment of extreme pleasure to welcome all of you for the lecture series organized by Department of Microbiology, Modern College of Arts, Science and Commerce, Shivaji Nagar, Pune. On the behalf of Department of Microbiology, I, Mrs. Madhuri Kothavde, Assistant Professor, offer my regards to all our guest speakers, Principal Professor R.S. Zundarao, Vice Principals, IQAC Coordinator Dr. Anjali Sardesai, Madam, Head of the Department Dr. Shilpa Mazumdar, Madam, my all colleagues, and all my dear students. In the current time, one of the principal public health problems of the 21st century is emergence of antimicrobial resistance, that is AMR, that threatens the effective prevention and treatment of an ever increasing range of infections caused by bacteria, parasites, viruses, fungi, and no longer susceptible to the common medicines used to treat them. So, considering this problem, this year our lecture series is organized on the topic of how to conquer the antimicrobial resistance, and it is entitled as Overcoming the Challenge of Antimicrobial Resistance. In this series, we'll be having guest speakers who will talk about the antimicrobial resistance and how to overcome this challenge in the current situation, in current scenario. And with this introduction, now I request Dr. Shilpa Mazumdar Madam to say a few words about the lecture series. Over to
Thank you,
you,
madam.
Shilpa ma'am.
Yes, thank you, madam. Good morning, all. Respected Principal Sir, Dr. Rajendra Zundara, respected guest speaker, Dr. Karishma Pardesim, Head Department of Microbiology, Savitri, dear students. I welcome you all for a two-day lecture series organized by Department of Microbiology. Every year, we organize one lecture series on important topic in microbiology to update our students with the current knowledge in that area. Last year, we could not able to conduct the same because of COVID-19 conditions. Although I'm happy to announce that we are conducting it this year, and the topic of lecture series entitled as Overcoming the Challenges of Antibiotic Resistance. Antibiotic resistance is emerging trend and we need to control it at priority. As per the reports of WHO and CDC, that is Center for Disease Control and Prevention in United States, latest national death and infection estimates for 18 antibiotic resistance bacteria and fungi. More than 2.8 million antibiotic resistant infections occur in US each year, and more than 35,000 people die as a result. India also reported as one of the highest rates of resistance to antimicrobial agents used both in humans and food animals. In India, the infectious diseases burden is among the highest in the world, and recent report showed the inappropriate and irrational use of antimicrobial agents against these diseases, which lead to increase in development of antimicrobial resistance. AMR has the potential to affect people at any age of life, as well as the healthcare, veterinary, and agriculture industries too. This makes it one of the world's most urgent public health problems. Although antibiotic resistance is a natural process, the main cause of antibiotic resistance is misuse of antibiotics. When we use antibiotics, some bacteria die, but resistant bacteria can survive and even multiply. The overuse of antibiotics makes resistant bacteria more common. A growing number of infections such as pneumonia, tuberculosis, gonorrhea, and salmonellosis are becoming harder to treat as the antibiotics used to treat them become less effective. Hence, there is urgent need to find solution for the same. With this background, we decided to organize this lecture series. In the lecture series, renowned scientists from this field will deliver lectures on challenges and solutions of antibiotic resistance. I'm sure students will be immensely benefited with this lectures series, and their curiosity will be satiated and deepened. I appeal students to take advantage of this opportunity. Thank you all.
Thank you so much, madam, for the introduction. Now I request our principal sir to guide about the session and inaugurate the lecture series.
Thank you, madam. In this inaugural session, we have vice principals of our college present. Heads of the departments are there. Resource person Dr. Karishma Pardeshi, Head Department of Microbiology of Savitribai Phule Pune University. Dr. Shilpa Muzumdar, Head of Department of Microbiology of our college. Mrs. Madhuri Kothaude, who is coordinating this particular activity. Other teachers from Department of Microbiology and other departments of our college, and dear students who are participating in this particular lecture series. It is a sort of, it has become a trend in modern college that along with regular teaching learning process in classrooms and in laboratories, this kind of activities are being conducted by almost every department. Let it be a lecture series like this, or a workshop, seminar, conferences, nowadays webinars. So we believe that through such activities, particularly when we used to conduct such activities offline, students used to get opportunities to work for organizing such events, and in the whole process, they were learning a lot of things about organizing certain events. They were also getting opportunity to interact with the resource person, and there are several examples where through such interactions students got some good opportunities in the field of research also. So such lecture series, webinars, conferences, seminars, they always help, and I wish to congratulate Head of Department of Microbiology of our college, Dr. Shilpa Muzumdar, and her entire team of very enthusiastic and active teachers for coming forward in organizing this kind of lecture series for the benefit of students. Now we are looking at such activities in interdepartmental perspective. Suppose this kind of lecture series, of course it is useful to other life science departments also. So the organizing department is contacting other allied departments, teachers, students, and encouraging them, appealing them also to participate. I am sure in this lecture series also, students, teachers from other departments have joined for taking academic benefit of this kind of activity. As a principal of a college, of course, I have to involve myself in admission process. So whenever students come for admission and they request for admission to microbiology, I used to ask them why you are particularly interested in microbiology. Is there any specific reason? And I used to get answer from these young students that, sir, nowadays we are facing several challenges due to Corona, COVID-19, and I want to learn microbiology, do some research, and find out some solutions or preventive measures for this kind of pandemic situations likely to come in future. So it is a good sign. Students nowadays, they think about the overall situations, think about the problems faced by mankind, and I want to do something. This kind of enthusiasm is observed among the student, and definitely the importance of subject microbiology has increased along with few other allied subjects, considering the challenges faced by mankind. As far as our microbiology department is concerned, very briefly, I am happy to mention here, this is the department which has very good collaborations with industry, They have been conducting pharmaceutical technique course in collaboration with some industries in and around Pune. Campus interview, though it is a traditional college, microbiology department always comes forward for organizing campus interviews, for organizing programs for employability enhancement for the student. So it's a very active and vibrant department. As far as university, Savitribai Phule Pune University is concerned, Department of Microbiology, we always have a respect towards departments on university campus. We always feel that these departments are playing parental role with respect to colleges affiliated to Savitribai Phule Pune University. As many of you know now, modern college has become autonomous. Last more than 2.5 years, we are successfully implementing autonomy, and our management, Progressive Education Society, has decided that in future, probably we would like to become modern university also. That might happen in next few years of time. Since we are autonomous now, we have freedom to design our own curriculum, to add some important aspects in the curriculum. And that's why on this occasion, I would like to appeal the resource person that if you would like to suggest later certain areas which we should include in our regular curriculum, or we should design some kind of add-on credit courses, we have the freedom as autonomous college. So do guide us in future with respect to this kind of possibilities, enriching our curriculum or designing some add-on courses or employability enhancement courses or some kind of add-on credit also. I am happy to mention here that we have received funds to the tune of 1.3 crores of rupees for establishing central sophisticated analytical instrumentation facilities. Such facilities are observed on university campus, in national laboratories. Very few traditional colleges have such facility. We have created this facility. Credit goes to Department of Science and Technology, DST, of Government of India. We have created that facilities and we have started using the facilities. With respect to that also, the resource person, Dr. Karishma Pardesi, later can guide our teachers from Department of Microbiology. So I once again welcome you all in this particular lecture series inaugural function. Once again, I congratulate Department of Microbiology for organizing this. I am happy to welcome the resource person, Dr. Karishma Pardesi, to Modern College again, and I express my best wishes for rest of the program. Thank you very much.
Thank you very much, sir, for inaugurating the series and guiding all of us. The lecture series will be conducted for today and tomorrow. So, student, you will be receiving the link for feedback form at 12:30 to 1 p.m. only for both the days. Students submit the form for both the days in order to receive e-certificate for the lecture series. So do fill the feedback form within the given time. Now, I wish this lecture series enlightens us and provides us with the approaches to deal with the problem of antimicrobial resistance. So now, without wasting our time, let's start with the first speaker of our series, that is Dr. Karishma Pardesi, Head, Department of Microbiology, Savitribai Phule Pune University, Pune. Now I request Mrs. Anushka Deve to take up the charge and start with our first speaker in the lecture series. Over to you, Anushka ma'am.
Thank you very much, Madhuri ma'am. Madhuri ma'am, I hope I am audible.
Yes, you are audible.
Yes. Thank you very much. First of all, I welcome all the respected members in the lecture series, and I welcome Karishma madam in the lecture series. It's an honor to introduce Dr. Karishma Pardesi. Dr. Karishma Pardesi is working as an associate professor and head of Department of Microbiology, Savitribai Phule, Pune University. She has teaching experience of around 20 years at undergraduate and postgraduate level. Her doctoral research was on role of plasmids from Acinetobacter species in horizontal spread of antibiotic resistance in clinical environments. After PhD, she extended her research interest to studying other bacteria, escape group pathogens, which are listed in the priority list of pathogens against which antibiotics are urgently needed. She is guiding 5 doctoral students. One of them has been awarded degree recently. Her research team is working on characterization of features responsible for horizontal gene transfer, biofilm formation, and persister mechanisms, which are the principal cause of growing antibiotic resistance and recurrent infections. They are also exploring alternative strategies such as nanoformulations or bacteriophages against escape pathogens, especially those causing wound infection. She has around 31 research publications to her credit. She has been part of research projects funded by various government bodies like DCUD, UGC, CSIR, New Delhi. She is an associate editor and reviewer for some of the most reputed journals for both national and international. Dr. Karishma Pardesi worked as COVID warrior from June to October 2020 at the RT-PCR based SARS-CoV-2 testing lab at ICER Pune. She also led the team involved in setting up a similar BCL2 and level center for molecular diagnostic and research. She is a member of Board of Studies Microbiology at SPPU and 2 autonomous colleges also. She is also member of Institutional Biosafety Committee, SPPU and DY Patil College, Pune. Dr. Karishma is a life member of various scientific communities such as Indian Women Scientists Association, Association of Microbiologists India, Association of Medical Microbiologists India. As we look at the biodata, Dr. Karishma Pardesi is the perfect speaker for our today's entitled lecture series, Overcoming the Challenges of Antibiotic Resistance. So let's request Karishma Madam to begin her most awaited lecture. Over to you, Karishma Madam.
Thank you, Anushka ma'am, for such a nice introduction. And I would like to thank the head of the department, Dr. Shilpa Muzamdar ma'am, and the entire team of Modern College Pune Department of Microbiology for arranging this particular lecture series in which I am invited. I am glad to hear from Jinzar Rao sir about the progress about the college and the different activities that are being carried out at the department. So without wasting more time, I would begin with the presentation. Let me share the screen first. Am I audible to everyone?
Yes, ma'am.
Is the screen visible?
Yes, ma'am. Your screen is visible.
Is it the presentation that is visible?
Not yet.
Not yet. Okay, just a minute. Now it will be. Now,
Yes, ma'am. The presentation is visible.
and are the slides moving as well? Are you able to see the slides changing?
No, slides are not moving.
Okay, just a minute.
Yes, yes, now they are moving. Yes,
Are they?
yes, yes. It shifted to the next.
So it's changing, right?
Yes, ma'am. Yes.
Okay, okay, okay. So thank you, everyone. The title of the talk that I've kept is No Escape. The spelling, as such, if you'll see, for escape is intendedly appearing wrong, but there's a reason for that. And the title is kept as a pun, which means both. Are we going to escape from the escape, or there is no escape from the escape? So let's see what all the escape is about. To begin with, what we'll do is we just introduce a few terms, starting with nosocomial infections. So, as the introductory session of this lecture series began,
Karishma madam,
yes,
we cannot see the title slide. We are able to see the fourth one.
okay. So, let me share the entire screen so that there is no hassle. Now, is it visible? The
Yes,
title
yes.
screen?
Now it is.
And now, is
Yes.
it changing as well? Is it?
No.
It's
Yes,
changing.
yes.
Okay.
Yes, ma'am. It is changing.
Okay. So that was the title of the talk, No Escape. And it, as I said, is a pun, intended pun to indicate whether we are able to escape the escape infections or we are not able to. To begin with, we just understand a little bit about the nosocomial infection. So, as the term means, it is an infection that is acquired by a patient when he or she is hospitalized. So, any infection that would be acquired by any patient who is hospitalized would be termed as a nosocomial infection. And these are one of the major causes of all the AMR or resistance or the antimicrobial resistance that we are seeing. These nosocomial infections, these are also termed as hospital acquired infection or healthcare associated infections because that's what they mean, that they are the ones that you acquire when you get admitted to a hospital for any treatment. So, as Shilpa ma'am already indicated, the CDC report, where they are trying to evaluate the uh, number of infections that are seen due to HAIs or the hospital acquired infections in US, they found that it was around 1.7 million hospitalized patients annually who acquire hospital acquired infections. And more than 98% of these patients are seen to die because of these infections. Now, this was a data that I have taken from the infection the drug resistance in 2018. After the COVID situation, it has been reported, although there are the figures are yet to come, all but there are reports which state that the number of antimicrobial resistance pathogens being reported is rising due to these COVID situations also. So there are quite many studies related to that. What would be the different kinds of infections that one may catch when he is in hospital? So it could be the surgical site infections, mostly the wound infections or the site which is operated. So it could be those sites that could get infected. It could be the UTIs, the bloodstream infections, some gastrointestinal infections, pneumonia, or any other infection that the patients would catch when they are getting in the hospital. So they are getting in the hospital for one particular treatment, but while being treated, they are catching these infections. The patients who are most likely to develop these infections, these are mostly the elderly ones who are not having a very prominent immune system. It could be the premature infants or newborns. Again, in them, the immunity is not well developed. It could be the burn patient or the surgical wound infections that would be there in the patients, people who are diabetic or who are suffering from cancer, or any other immunocompromised persons. These would be the ones who would be most likely to catch the hospital acquired infections. And sometimes it would also be people who are undergoing dialysis or catheterization when they are at the hospital, because of which again they would be quite likely, quite prone to catch such hospital acquired infections. How would these people catch these infections? So it could be a variety of sources from which the infections could be caught. So you see the schematic of a hospital over here and the different sources which contribute to the hospital acquired infections. It could be coming from the patient who is suffering from the infection and he is spreading the bacteria when he is undergoing treatment at the hospital. The hospital personnel or the staff who is treating the patient is likely to visit the patient and is either transmitting some microbes to the surrounding of the patient, or is carrying the bacteria when the hospital staff has treated the patient. It could be the equipment that were being used to treat the patient that could get contaminated. It could be any other surfaces in the hospital, maybe the elevators or the floors or the trash bins or the ventilation ducts, the AC ducts that would be there. These could contribute to the infections. So probably the sources that you could see would be any of them. It could be inanimate or animate sources, or it could be such surfaces, and the people who are moving from one ward to the other, they would be transferring such infections. There would be a variety of organisms which are involved associated with this hospital acquired infections. These could be the gram positive bacteria, the gram negative bacteria, fungi, as well as viruses. I've listed a few of them. So there are some 17 to 18 pathogens that are most commonly associated with these HAIs. And some that have been marked in bold are more critical or more significant or more clinically important to be studied because of their MDR nature. So if you just uh, read the first letters of this, they would try making sense to the title of the talk that I gave. So these are the escape pathogens, and they are responsible for majority of the hospital acquired infections or the nosocomial infections. Why? Because they are able to escape the biocidal action of the antimicrobial agents or the antibiotics that are used to treat them. And this term, it was coined by Rice in 2008. It is an acronym for the 6 pathogens which are majorly involved in all the AMR that one would see around, or all the HAIs that would be contributing to the hospital infections. So these are Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumoniae, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. So that's how the name arises, ESKAPE, escape. And since they are able to escape the biocidal action of the antimicrobials, the acronym is perfectly suiting those pathogens. Most of them are multi-drug resistant isolates, and they have several other features which make them uh, kind of powerful in surviving in the hospital environment and their ability to cause infections. So it is very important to understand the resistant mechanisms of these bacteria and to also develop novel antimicrobial agents or other alternative tools to combat such public health challenges. So why these escapes are important? These escapes, they have a variety of features. One, they are nosocomially associated. They are predominantly present in the hospital environment. So of all the other pathogens, the escape group seems to be most commonly occurring in the hospital environment. Most of them are multi-drug resistant to a variety of classes of the antibiotics. They could belong to beta-lactam, the carbapenem, cephalosporins, tetracyclines, aminoglycosides, quinolones. So you see almost all classes of antibiotics are covered by these escape pathogens. They are resistant to most of them. They also have several virulence features which make them cause the infection or which make them survive into the environment. These include the exopolysaccharides that they produce, some hemolysins, the phospholipases, and two major virulence features. One is the biofilms, which are the major contributor for the recurrent infections, as well as the persister state that these organisms can get into, which also contributes to the recurrent infections. So we'll talk more about the biofilms and the persister state, which is a major threat in the hospital associated infections. And lastly, these escape pathogens, they also have ability to transfer their resistance traits to other organisms, to transfer their virulence trait to other organisms. Why? Because they have several mobile genetic elements. These could be plasmids, these could be transposons, these could be some other integrons, which would allow the resistance genes or these virulence genes to be transferred from one bacteria to the other. So gene transfer is one more way by which the resistances or these virulence features they can spread within the different types of bacteria. The first one, as I said, the biofilms, these are one feature or one virulence trait which makes the escape pathogens very powerful. So what are biofilms? So these biofilms, these could be the ability of the organisms to colonize different surfaces, which could be biotic or abiotic. I've just taken an example to show how the biofilms are formed. So there would be a bacteria which would adhere or start colonizing onto a substrate. They would start surface conditioning the surface on which they are to form the biofilms, slowly start multiplying, increase their numbers by replication, grow, and other bacteria, they would start coming towards the site where the biofilm is being formed or that microcolony is being formed. These microcolonies, they would form an exopolysaccharide cover around them so as to protect the biofilm, and this would lead to formation of a mature biofilm, which is going to be a colony of organisms which could be of different types. So it's not just one single bacteria, it could be a variety of bacteria that could together form biofilms, and such mature biofilms could then allow additional areas or other additional microcolonies to be formed by releasing the cells into the environment and allowing more biofilm formation. So this is typically seen in a variety of areas. The catheters, you would see that the biofilms are being formed. The dental implants that people get themselves. So there the biofilms are formed. It could be the contact lenses. So you would see that the biofilms could be formed over there. So these are some of the common places where you would see, or the common um, areas where you would see that the biofilms are going to be formed. The other mechanism which is involved in the spread, as I said, is the gene transfer. It could occur both by horizontal or a lateral gene transfer process or a vertical gene transfer process. A schematic to just explain the horizontal and the lateral gene transfer, and the vertical gene transfer has been indicated over here. So transfer of genetic material from one bacteria to the other directly, it is what you understand by a lateral or a horizontal gene transfer. This is how majorly the resistance genes would be transferred, or this could be the major cause because of which the resistance is spreading. And vertical, it is just the transfer of the genetic material from its ancestor to the daughter cell. So from the parent cell, the daughter cells arising, they are going to carry the same genetic material as the parent cell. It could be the transfer of genetic material in this order, but the lateral or the horizontal gene transfer is one of the major contributors. And Apart from that, so the organisms, they were resistant to a variety of antibiotics, but there are certain bacteria which are not resistant to antibiotics and they can still persist in the environment. So these are the bacterial persisters, which are again one of the major causes or major problems that one has to treat because they result into recalcitrant infections. So this graph on the right side, it just indicates the different types of bacteria that one may find. The antibiotic, when you are introducing the antibiotic, you would see that the organisms that are there, they could show different responses to the antibiotic. If the organism is resistant to the antibiotic will not have any effect on the organism, and the organism will happily grow, or it would not be killed in the presence of the antibiotic. So here this blue colored line that is indicated, it is just to show that whenever a bacterial population is exposed to antibiotic, if the antibiotic is not having any effect on the organisms, they would not show any change um, in their numbers. If the organism is susceptible to the antibiotic, then you would see a sudden decline in the number of cells in the log CFU value, and that is the dark gray colored line to indicate that the population is susceptible to the antibiotic. It is being killed in the presence of the antibiotic. There are some organisms which behave in between these two traits. They are neither resistant nor they are really truly susceptible. So these are the ones which are either the persister or the tolerant cells. The tolerant cells, they are slowly killed in the presence of the antibiotic. So this steep decline that you see in the log value in a time frame is going to be more slow if the organisms are tolerant to the presence of the antibiotic. Whereas persisters, these are the bacteria which are similar to the tolerant, but they are still very slowly being killed by the presence of the antibiotic. You would see a typical biphasic curve in organisms which show a persister state. So these bacteria, they are actually not resistant. They are not turning resistant to the antibiotic, but they are just becoming kind of dormant or silent. They are reducing their metabolic activities, because of which they are no longer able to respond to the effects of the antibiotic. So this becomes a problem because you are treating the organisms with antibiotic. This organism is not growing itself, it has gone into a dormant state, so the antibiotic is having no effect on it. And when you remove the antibiotic, the organism starts multiplying. Because there is no antibiotic around it, it would start multiplying. So the basic trait of the organism is going to be sensitivity to the antibiotic, but it would go into a dormant state and it would survive, it would persist over there, so the name persisters. So this is a major problem, and that's the reason why WHO has listed quite a few pathogens in the list of critical pathogens against which antimicrobials are required, or against which newer ways of treating them are required. And if you see the escape group, they come into the top priority list, the priority 1 and the priority list, or priority 2 list, that is the critical priority and high priority pathogens against which the antibiotics are required. Why? Because Acinetobacter baumannii, mostly these are shown to be carbapenem resistant, the carbapenem resistant Pseudomonas aeruginosa, carbapenem as well as ESBL resistant Enterobacteriaceae group, or the vancomycin resistant Enterococcus faecium, or the methicillin or vancomycin intermediate resistant Staphylococcus aureus species. So these are the different classes to which mostly these are found resistant, and that becomes a major problem to treat because we are having very few antibiotics to treat such pathogens. So this table is just indicating the 6 escape pathogens, the variety of antibiotics that were recommended by CLSI to treat such escape pathogens. But if you see this table, the red colored uh, rectangles, they are indicating that although those antibiotics were present earlier before 2010, after 2010 they started being removed from the CLSI guidelines. Why? Because most of these staphylococcus species, they started showing resistance to these antibiotics, and that is why the CLSI stopped recommending them for treatment. The ones that are shown in orange or the yellow color one, so these are the ones against which they were reported as the possible antibiotics to treat different escape pathogens, but the resistance was noted against them. And the green ones was a data which was to show that against these antibiotics, at least there are no reports of ephesium or S. aureus showing any resistance. So at least these antibiotics will surely work if the organisms are resistant to any other antibiotics. It's not that all the organisms in the world are going to be resistant to these antibiotics, but most of them are, and that is why those antibiotics are not being recommended by CLSI. So if you're seeing the chart, it would see very few green colored rectangles, which indicates that we are not having a wider choice of antibiotics to treat such escape pathogens, which is again a cause of concern. And so we need to have alternative ways to treat the escape pathogens. There are variety of alternative therapies that have been reported. I have listed out some 3 or 5 of them, and I'm just going to be talking about 3 which are given in bold. So it could be, if single antibiotics do not work, then you can use antibiotics in combinations or you can use them with certain adjuvants. It could be the bacteriophages that could be used as alternative therapies. Why? Because antibiotics are no longer working. It could be antimicrobial peptides or photodynamic light therapy or the nanoparticles. So these are few of the alternative therapies that have been listed out. There are quite many more which could still be used. However, a lot of work, a lot of research needs to be done in these areas. So the first one is the use of antibiotic combinations. As I said, instead of using one antibiotic, if you use 2 antibiotics, it could show any of the 3 responses or even 3 major responses. There could be some other responses as well. So the 3 major bacterial responses, when you are exposing them to combinations of antibiotics, you would see that the effect of the combination could be additive, it could be synergistic, or it could be antagonistic. I'm sure you would be slightly aware of these terms. Additivity is going to be when you are having the antibiotic, both of the antibiotics added in increasing concentration, you are not seeing a pronounced effect as compared to the individual antibiotics, but it is not at least declining as you see in the antagonism. In synergy, the combination decreases the MIC or decreases the amount of antibiotic that would be required to kill the pathogen. And antagony, it is when the 2 antibiotics are taken together, they are not complementing each other and they are instead acting to allow growth of the organism at the concentrations that were otherwise the killing concentrations. So these uh, stars or these pointers over here, they are to indicate that those would be the concentrations that would be the killing concentration or the MIC concentration of the antibiotics. If you are using both of them together, then the antibiotics are going to be used in lesser amount. The number of organisms are going to reduce, but they are not going to cause complete killing of the organism. In synergy, the killing would happen at a much lower concentration in the combination. So if the concentration for antibiotic A was 100 micrograms per ml and concentration B for the other antibiotic was 50 micrograms per ml, in the combination you may not require 150, you may require just 25 and 10 of each of them. So that's how the amount of antibiotic required is less. The killing of the or combination is going to be more pronounced. And this is just a schematic to indicate when you are using either individual antibiotic alone, you would see how would it affect the culture. It's causing your, in this case, they are not showing all the organisms gone. So the individual antibiotic probably is not having any effect on the organisms. You are seeing more number of cells, but when you are using them in combination, you would see a difference in the growth of the bacteria. It could be additive, at least declining but not completely killed, synergy very prominently being killed, or antagony not being killed at all, instead the numbers rising. What would be the mechanisms by which these antibiotics are causing such synergies? So that is indicated in this particular slide. The upper panel is just to indicate how the antibiotics are going to be acting. So the different antibiotics belonging to the different classes, and what would be the target that they are going to be looking for when they have to attack the cell. So there are organisms or there are antibiotics which are going to attack the cell wall or the cell membrane listed here. Some may attack the ribosomes and affect the protein synthesis. Some would affect the replication of the DNA, binding to the DNA gyrase or affecting the DNA gyrase. Or there could be other ways by which these antibiotics are going to be acting. So mostly the cell wall, cell membrane, protein synthesis inhibitors, or the nucleic acid inhibitors. So affecting the major functions of the cell. When you talk of the antibiotics used alone, probably the antibiotic is not able to enter the cell. Probably the protein synthesis is not being hampered because the site where the antibiotic has to act has been modified by the organism, and the organism is not able to, the antibiotic is not able to recognize that site, because of which it's showing antibiotic resistance and the protein synthesis is not being affected. So when you use a combination, you would see that one antibiotic would complement the other in variety of ways. It could be uh, inhibition of the the agent that is otherwise interfering with the activity of the antibiotic. So penicillin and clavulanic acid combinations, you would see that clavulanic acid is the beta lactamase inhibitor. It will try to inhibit the beta lactamase, which is the enzyme which is going to act against this antibiotic of the beta lactam groups. So beta lactamase inhibitor is added to inhibit the action of the beta lactamases, and so penicillin can act. And likewise, all the other combinations, they could help in some or the other way, complementing the other antibiotic, or uh, helping the other antibiotic to either enter the organism and cause the killing effect. That could be one of the way, the combination of antibiotics. But as you know, we have little combinations or little options available for the different combinations to be used. So still there is not really a good achievement that you don't have enough of combinations against which the escape are going to be treated. So the other alternative therapies that could be thought about would be bacteriophage therapy. This is just a indication to show how the bacteriophage therapy that initially began, it got lost when the antibiotics came into existence. So the initial therapies that were being used, bacteriophage therapy was being reported as one of the ways to treat bacteria. So in the early 19th century, you would be seeing that bacteriophage therapy was being used. But as soon as the antibiotics were introduced, the era of the antibiotics came, you saw that there was a decline in the use of phages being used as therapeutic agents. And slowly you started seeing the rise in the antibiotic resistance traits shown by the organisms. So the uh, use of phages again started increasing, or it started improvising. Why? Because the options of antibiotics became little and little. So the decline in the initial phage therapy was due to the advent of the antimicrobial era, and as you saw the rise in the antimicrobial resistance, again we are going back to the olden days, olden therapeutic agents like phages. How would phages act? So the conventional phage therapy, it would be like you have a phage which is having an ability to to attack or to infect a bacteria, and you have such phages isolated, prepared in large numbers, and you could use it against pathogens to uh, which those phages can infect. So it could be the phage that could be used as a single monophage. which is going to be against one bacteria, the phage is going to go infect the bacteria, and it is going to cause the bacteria to lyse, to be killed. The phages are going to be synthesized in large numbers, and they would in process kill other pathogens around them. So that's how the bacteria would get killed. It could be the phage derived enzymes that one could use. So the phages as such directly, instead of using the phages, it could be the enzymes that are used by the, or that are synthesized by the phages, which are helping in lysing the bacterial cells. These could be uh, removed, or these could be purified, and then you could use those purified products of the phage against the pathogens, and you could treat the pathogens. It could be combinations of phages with some other antibiotics or antibodies which could be used against the pathogens. So the phages are going to create a kind of hole, or it's going to create a kind of entry into the bacteria, and the other antibiotics which were otherwise not able to enter into the bacteria get entry via the phage into the bacteria, and it is able to kill the bacteria. So this could be one of the mechanism, or it could be bioengineered phages that could be used, which are having some antibiotics or some photosensitizers bound to them, or en- engulfed in them, and these are going to be uh, used for treatment of the infections. So phage therapy, it could be used in a variety of ways. A lot of research is being going on to isolate, because when you say the phages have to, they are going to be infecting a bacteria, you need to have phages against all possible types of pathogens that are uh, there, or that may be causing an infection. So the major challenge as such in phage therapy is going to be to have a phage library, or to create a collection of phages against a wide variety of bacteria. So this is just a flow chart to indicate how would phage therapy be actually be brought into clinical practice. So one would have to isolate phages against a variety of bacteria which are known to cause infections. Such phages need to be stored as a phage library. Effective phages against the pathogens. If a person is suffering from any bacterial infection, then an effective phage should be provided in high dose to that person. But before this phage is being used, it needs to be uh, prepared in a manner which is going to be safe to administer. So there are going to be safety studies that need to be done. Then the phages need to be present in high titer, and they should be pure. They should not have any endotoxins or any other impurities which may not be very safe for administration into humans. So those safety studies, toxicity studies, they need to be carried out. Only then such phages could be used against the bacterial infections that the patient is suffering from. Alternative way could be, it could be a therapy to have, first you have the patient, you don't have any phage for this particular bacterial infection that the patient is suffering from, so there is no phage in the phage library. Then in that case, it would be, you isolate the bacteria that is causing infection, you identify that bacteria, you get that bacteria to the phage lab, and the phage scientists over there, they are going to isolate a phage against this bacteria, and then prepare a formulation which is going to be administered to the patient, and it would help cure the patient. But this process appears to be very long, that after the patient has suffered from infection, the antibiotics are not working against them, then you go in for isolating bacteria, then you provide that to the phage lab. The phage lab is going to isolate a phage, and then it is going to provide the therapy. So it's kind of a therapy which is targeted therapy, or it is a therapy, personalized therapy kind of thing, which is provided, but the time taken is going to be more for this. And plus there are going to be possibilities that these phages are going to show resistance to the bacteria. So then again, you'll have to find some other alternatives or some other phages, or you need to have a variety of phages against the bacteria. So general protocol is going to be a pretty long protocol. You need to have safety studies created, you need to have such phage library created, and you need to have phages against all possible pathogens that one might be expecting to cause infection in patients, against which you can provide these phages. So that's kind of difficult, but still people are going by this, or they have begun carrying out such preparing such phage formulations and providing them. So basically, as I said, the phage therapy, three aspects that would be required to select the phages would be: these phages should follow only the lytic cycle, or they should be particularly engineered phages that you'll be using them. They should follow a lytic cycle, because if they enter lysogeny, then those phages are going to get into the host or the bacterial genome, and they are going to add and or carry the genes from the bacteria, and those genes could be other resistance gene. So they would rather contribute to the spread of resistance. So you don't want that. You want only the lytic phages. They should not enter into the lysogeny state. Those phages that you select, the host range has to be checked, and such phages should be at least having a wider host range, or you should have many phages that would have different hosts. You could use them as a cocktail of phages. You need to study some make some stability studies or temperature pH stability studies, because you're going to make a formulation of those phages, and you have to see whether those phages, it was a in vitro study that you have carried out, so you need to see whether these phages have an ability to remove the target bacteria when they are at the site, maybe in biofilms or in sputum or in cell lines or in some wound. So you have to carry out some ex vivo in vivo study to see whether those efficacies are really observed in invertebrate or animal models. And you also have to carry out some safety studies, as I already indicated, to show that these phages, when you are treating them, they are not harmful to the person who is receiving such phage treatment. They should lack immune response. They should not show any immune response. These phages should not generate any immune response, which is going to be harmful to the person. So these would be the major things that one would look at when they are selecting phages for any therapeutic purposes. The last one is the antimicrobial mechanisms or the nanoparticles that could be used. So this slide is just to explain the nanoparticles or the nanomaterials that could be used against bacteria. Nano particles or nanomaterials, you would say these are any organic, inorganic molecules that could be used against organisms, or that could be of a size which are less than 100 nanometer, and these could be used for different applications, either in agriculture or in medicine or in for environmental purposes, or for any other biosensor applications or anything. This particular slide is just to indicate the antimicrobial mechanisms of nanomaterial, to show how metal nanoparticles or some other the carbon based nanoparticles, these could be used for treatment of infections. So metal nanoparticles, these could be either prepared in some liposomes which are going to carry those metal nanoparticles, or some nano emulsions that could be made out of them, so that these would fuse with the bacterial membrane, and they would allow the entry of those nanomaterials inside the bacteria. When inside the bacteria, these nanomaterials would act to affect the different target sites, either the different intracellular components of the cell, like the ribosomes or the DNA, and interfere with the protein synthesis or the DNA replication. They could generate an ROS response, or simply they could be used for delivery of some other therapeutic agents inside the bacteria. So they are acting as the delivery agents. So one, it could be those nanomaterials or the nanoparticles themselves are acting as agents to interfere with the bacterial uh, mechanisms, either the protein synthesis or the nucleic acid synthesis, or they could act as delivery agents where they are carrying some other uh, antibiotic or maybe some other therapeutic agent inside the organism and helping treat the bacteria. So this is one more slide to indicate the metal nanoparticles that could be causing harm to the bacteria or causing the killing in the bacteria, where it could generate an ROS response. It could kind of affect the protein synthesis, or it could inhibit the efflux pumps which are effluxing out the antibiotics and making the bacteria resistant to antibiotics. So you could have the efflux nanoparticles coming as efflux pump inhibitors. They are blocking the efflux pumps, and therefore they are allowing not allowing the antibiotics that are added to be effluxed out. So the antibiotic and the nanoparticle combination could be used as a combination to treat such bacterial infections. So again, the strategies that could be used to explore the nanoparticles and their and their medical applications would be have different types of nanoparticles used for monotherapy, or they could be used in combination, or they could be used in conjugation. You need to carry out several cytotoxicity studies before you actually get them into practice, both at in vitro level as well as at in vivo level. So in vitro level, you just try to see using different implants whether how these nanoparticles are going to be acting, whether they are able to generate any allergic reaction, whether they are aggregating and they are losing their stability. So all such studies maybe you would have to do in vitro level. And finally, in vivo level, you'll have to see in animal models, you'll have to see how would it affect when the organisms or that animal is infected. So some infection model studies need to be done to explore the possibility of those nanoparticles being used for medical applications. Again, studies such as this, these are limited only to showing those nanoparticles are having antimicrobial property. There may be studies which show that they are safe to use, but as such, these infection model assays or actually being used in therapy, there are very few studies reporting that, and that's kind of a limiting step, because of which nanoparticles, although they are their application or their importance or their use for treating bacterial infection is shown at in vitro level, but advanced studies to use them in infection models or to actually carry out trials using such nanoparticle formulations, you would see a very handful studies related to this area. So many more studies, at least the toxicity studies and the animal model studies, they are required in order to really prove the efficacy of these nanomaterials being used in therapy. So the major challenges as such, when you're using antibiotic combination, we see that those antibiotic combinations could be antagonistic, they are not going to function. Phage therapy, I explained you all that the organisms could be susceptible to a phage and you could use that phage against the bacteria, but sometimes the bacteria, they develop phage resistance. That is, the receptor where the phage is going to be binding and it's going to allow the injection of the DNA into the host, that is going to be blocked or is going to be modified, therefore the phage will not be able to enter, so the bacteria is becoming resistant to phage. This is going to be one of the problem in phage therapies. In nanotherapy, these nanoparticles or those nanomaterials, many of them are mostly unstable, they aggregate, and such aggregates will not be able to enter into the cell membrane. So even though the nanoparticles were formed at an in vitro level, you saw that they were used but they were not stable, because of which they could not gain entry, or sometimes they are harmful to the host cell, or that is the human cell or the animal cell, because of which you cannot use, they are not specified only to the bacteria, they could be showing the same effect against the human cells also, because of which you could not use them. So this is again a major limitation. Antimicrobial peptides and other combinations, I'm not talking about that, because we have not used them in, or we have not discussed them in the current presentation. So, what could be the alternative? If monotherapies are not working, the alternative could be we could use those monotherapies as combinations. So, when you say that nanoparticles are not able to function, but nanoparticles may function when you're combining them with either some antimicrobial peptides or maybe some other phytochemicals or some antibiotics. So, nanoparticles are not to be added in very high concentrations. If they are present in high concentrations, then probably they aggregate or they are affecting, they are having toxic effect on the host cell as well as the bacterial cell. So instead of that, you use them in combination with antibiotics, so the amount of nanoparticles required is going to be less, and then the antibiotic can easily act, or those nanoparticles can create entry points so that the antibiotics can gain entry inside the cell. Same way phages could be used, where you club the phages, or you have some engineered phages made, which are combination of nanoparticles and phages or antibiotic phages combination, and use them for drug delivery. Or it could be any other combination of these monotherapies that could be used for treating the infections, the escape related infections. So these are the different possible ways by which the escapes could be treated. Finally, if the limitations are there, then prevention is the best way to treat such hospital acquired infections, majorly the escape infections. So we have been quite aware of this due to the COVID situation. Many of these you would be practicing now. So having educated hospital staff who is following hygiene in theater, the operation theater, the wards, the kitchens, following proper surgical techniques, frequent hand washing, proper sterilization or disinfection of the equipments that are being used for patients, then creating isolation wards for infective patients, or taking extra precautions for immunosuppressed patients, limiting the use of antibiotics, keeping a constant surveillance of infections in the hospitals. All this you can relate to the COVID situation also. So, same way, the nosocomial infections could be controlled. So, it's better to control them, it's better to prevent them rather than finding strategies to treat them, because you're seeing that there are quite many limitations, or still a lot of work needs to be done. It's not that I'm not giving a false hope that um, it's a very nice way to treat the pathogens when you have all those alternative therapies. They have limitations, although we are advancing in research to treat such infections using those alternative therapeutics, but some more work, some more research is required for that. Lastly, I would like to give a brief introduction to the research that is being carried out in our lab. So, this was already mentioned in the introduction given by Anushka ma'am. We are studying the mechanisms of drug resistance in escape pathogens. So, it could be the persister cells, it could be the different antibiotic resistance traits that these escape pathogens are showing. So, what is the reason why these escape pathogens are resistant to so many antibiotics? It could be efflux pumps. So we are targeting to study the drug resistance mechanisms of escape pathogens. We are also studying the virulence traits in these escape pathogens, particularly with relation to the biofilms that they form. So how is it possible to treat these biofilms using those alternative therapies? Then one of my students is also working on the mobile genetic elements which are involved in the horizontal spread of resistance. She's taken particularly the Acinetobacter species, one of the escape pathogens, to study these mobile genetic elements and see what are the variety of mobile genetic elements found in escape pathogens, one of them, and how would it help in spread of the resistance from one bacteria to the other. And lastly, we are looking at different alternative strategies, particularly to treat wound infections against these escape pathogens, in which we have targeted the PARG based therapies and nanoformulations or combination therapies for being used against such escape pathogens. Although our research is more at an in vitro level and few animal model studies that we are projecting to do now, so a lot of work is still there, a lot of scope is still there to be done. This is my research team. I have five PhD students who are working with me. So Sheetal, Ekta, Madhumita, Mansura, Suresh Basutkar sir. So these are the five who are working with me for PhD currently. Ekta ma'am is already known to you, and she has been awarded her PhD degree on the persister cells topic that she worked on. Apart from that, I do guide dissertation students both at the department level as well as coming from other colleges, and they have been there carrying out the research that I do at this lab. So these are pictures of few of my dissertation students who have been working with me and who have contributed to the research that is going on in the lab. This is Ekta ma'am, who was awarded the PhD degree last year, and that's all from my side.
Thank you very much, madam, for such an informative lecture and hopeful lecture, like the strategies which we might explore ahead to escape the challenge of antibiotic resistance. I request audiences, if they have any questions, they can put in the chat box, or maybe they can ask directly to speaker. If anyone have query, just post it or maybe ask
Yes,
directly. Yes, Mohit. Yes, go ahead.
ma'am, actually, I have two doubts for the madam. Can I ask?
Yes.
Yes,
Yes, you can.
Yes, ma'am. Thank you. Ma'am, while studying the medical microbiology at college, we learned that one more reason for this antibiotic resistance was the prescribing the wide spectrum antibiotics by the practitioners and not diagnosing the disease causing organism to their species level. And for
yeah.
identifying the organisms, molecular techniques like PCR are used, which are not accessible at the ground level practitioners. So, ma'am, my question was that is there any recent methods and scopes designed or under development to provide the cheap diagnosis replacement to this high cost molecular diagnosis?
Can you just repeat the last sentence that you said, the question part?
So, ma'am, my question was ma'am, is there any recent method and scopes designed or under development to provide the cheap diagnosis replacement to high-cost molecular techniques like PCR?
See, now the problem with the high cost, your question was, ke, uh, we are prescribing antibiotics rampantly. Uh, a lot many antibiotics are being prescribed without even knowing what the organism is. So it's like you are treating the patient before you start getting the culture report. Many of these infections, you don't even ask the patient to go in for a culture of that sample, and you start prescribing antibiotics based on a previous knowledge that so and so infection is usually caused by these organisms. So UTI infections usually it would be a Klebsiella, it could be a E. coli that could be causing that infection, and accordingly you would have antibiotics prescribed without even knowing whether the Klebsiella strain or the E. coli strain causing the infection is indeed susceptible to the antibiotic that you are using. And that this is a major cause, as you rightly said, for treating for the rise in the resistance that we see. The alternative to this could be you have ways or methods where you can long term, because the organisms take a longer time to grow. But these days, and that's the reason why you're not able to you cannot wait, and you have to go by prescribing the medicines before the culture report comes in. So the other advanced therapies, like advanced methods, diagnostic methods like the PCR based methods or some other biosensors, these could be used. Some kit based methods, these could be used to identify the pathogens. Although it may not be possible to have methods for all of them, but there is a lot of research going on to have sensors designed where you can do some rapid tests to identify pathogens. There are ways by which you can rapidly identify organisms, like it's not the conventional way you do a typical biotyping and identify the organisms, but you can identify them by using protein based methods, or you can identify them by the biotyping cards that you get. So this would make it easier, faster to get the identity of the organism. So yeah, the studies are, or the research is moving into direction where you are having diagnostic methods which are faster to help identify pathogens, although not all pathogens we know have such kits developed, and so you still go by the older ways of prescribing. Plus not all the hospitals would be so advanced having such facilities to have the infections treated. So this is one major problem, and that needs to be addressed.
Yes, Mohit, you had one more query, or
Yes, ma'am. Thank you for the
yes,
first answer.
yes, go on, go on. You can
Ma'am,
answer.
yes, ma'am. Ma'am, one article from NCBI, they had stated there one statistic. They said that in America, the 18 pharmaceutical companies which were major for antibiotic generation, antibiotic designing, out
Mhm.
of 18, the 15 antibiotic companies had pulled out from the business. So, ma'am, in very general, what are the major industrial challenges for developing new antibiotics? And as as they are like these practitioners, they are specifically they are reserving this new antibiotics resistant by the organisms. So what are the specific challenges faced by the producers for producing a new antibiotic?
The cost, I would say, the time taken to carry out trials and go through the different trials and then get them. So usually if you search, I did not put that slide in my presentation, but if you search, you would see that what would be the timeline that would be taken for development of any antibiotics. So they have put up, like when you just put in this the timeline for antibiotic development in Google, you will see that they give that the time that is taken for any antibiotic to be introduced into the market is very slow. The time that the organism develops resistance to those antibiotics is going to be within few years. You'll start seeing that the organisms, they are starting to develop resistance against the antibiotics. But with the same pace, we are not able to introduce new antibiotics into the market. Why? Because the cost of development of the antibiotics, the cost of finding new antibiotic formulations or new antibiotic molecules is very high. The time taken to find them and to study them and to get them into all those clinical trials and being passed, it is going to be more. And that's the reason why majorly you would see people or companies falling out from that particular part. And that is the reason why people are moving towards the older methods, or moving towards alternative methods for finding ways to treat these pathogens. So yeah, the cost time factor is major, and we are not able to catch up with the pace of the organisms, I can say. The organisms are faster developing resistance. We are not able to introduce new antibiotic formulations with the same pace. So it's kind of a war between us and the organisms. Who wins, we don't know. So that's why, again, no escape was with a question mark also, whether we can escape them or they can escape from us.
Yes, true. Uh, Thank you very much, madam. Thank you, Mohit. One more question in the chat box. Is there any technique which is used to characterize nanoparticle interaction with DNA? Shruti Hire asked the question.
Yeah, there are several methods that are used to characterize the nanoparticles. These are some assays that one would do to see whether, initially, when you are trying to study the nanoparticles, like what we do at our lab level, we would be taking the nanoparticles, we would be treating them with the organism, with the bacterial DNA, and we'll be trying to see whether, when you are treating with the bacterial DNA, usually the DNA band, how you get, whether you are getting a trailing in that band, then probably it has affected or it is causing harm to the DNA. Plus, you'll have to carry out some other studies where you kind of gene expression studies that you could carry out, where you'd start seeing how which enzymes would these nanoparticles be affecting, and if those enzymes functions have been upregulated, downregulated, based on which you will try to understand what are the ways by which or what is the action of the nanoparticle on these organisms.
Yes, I hope Shruti, your doubt is resolved. One more doubt by Omkar Pandit. He writes, are there any therapies carried on the large scale using bacteriophages in India or any clinical trials? Actually, ma'am elaborated on this, but still,
Yeah.
if ma'am you can answer.
So bacteriophage therapy, if you're seeing in the Russian country, it is being used. It in some other parts of the world also it has started being introduced. However, in India, we do not have, like, it's like not an approval given of FDA approval given that okay, so and so phage formulation can be used for therapeutic purposes. But yes, recently there was a conference in Abhidanandar College where a professor he had explained, he has begun in Banaras Hindu University, he has begun a phage bank sort of, where they are having phages against different pathogens, and if any patient goes to them, it is a case by case study. If there is any patient who is not being treated by any modern day antibiotics, if going to them, then they are providing patch formulations and they are helping the patient treat the infection using the patch formulations. Although it's going to be a case study kind of work, they have taken some ethical clearance for this and they are carrying this out. But clear, open kind of approval given is not yet given for any of the phage formulations in India. Although there are studies which are showing that these phages could be used. So in poultry or in agricultural areas, people are developing phage formulations which could be used for medical applications. But no patch
Yes.
formulation as such has been given approval by the Indian Medical Association. But yeah, as a personal therapy, people have begun to use that, but it is used as a last resort. When antibiotics are not working, only then pages could be used. ऐसे करके they have used it.
Yes, such a overwhelming response by audiences. Students are posting questions, but we are running out of time. But still, ma'am, could vaccine be the alternate way to treat antibiotic resistance?
I won't say vaccine could be the alternative way to treat antibiotic resistance. Yeah, but vaccines are being developed against bacterial pathogens as well. There are a lot of studies which report development of or trying to study if vaccines or antibodies could be developed against the escape group pathogens also. But yeah, it's not yet done. They are there. There are studies reported.
Yes, thank you very much, ma'am, for resolving questions raised by students. Actually, there are many more. Ma'am, if you allow, we can share maybe your contact details so that they can approach you to resolve many more queries ahead. Um,
No
Ma'am,
problem.
in her lecture, she started the lecture with app titled No Escape to emphasize the importance of awareness about escape group pathogens. Otherwise, there is no escape from this dangerous situation. Ma'am also described about nosocomial infections and reasons to catch those. Ma'am also elaborated issues of emergence of antibiotic resistance and the solutions over them as alternative therapies like bacteriophage therapies, combination approach or nanomaterials, and even the challenges faced upon their applications and trials. I'm sure our students must have influenced and maybe chose their topic of interest in similar lines to pursue further. Thank you very much, ma'am, for enlightening us with the latest, the current information about the topic and making us aware about the research or the desire of research in this issue. Now I request all the participants to turn on their cameras for a few seconds so that we can have one picture with our respected speaker.
Thank you so much.
Participants, please turn on your cameras for the picture. Thank you very much. Thank you very much, Karishma madam.
Thank you,
And
Shilpa.
now over to you, Madhuri madam.
Yes, thank you so much. Thank you very much, Karishma madam, for the wonderful session. And thank you, Anushka madam and Jayashree madam. Now let's start with our second session for the series by Dr. Sidharth Chopra. I request Dimple madam to take up the charge and start with the session. Dimple madam, over to you.
Yes. Thank you so much, Madhuri ma'am, for giving me this opportunity to introduce today's guest speaker, Dr. Sidharth Chopra sir. So, sir has completed his PhD from ICGB JNU in 2004 with Professor Anand Ramanathan and moved to Stanford University School of Medicine, Stanford University, USA, with Professor Gary A. Skully till 2008. And from 2008 till 2012, he worked as a research scientist at SRI International Menlo Park, USA. In 2013, Sir moved to CSIR CBRI as Assistant Professor, Department of Microbiology, and has established a well-funded, vibrant laboratory working on drug discovery targeting MDR bacterial pathogens. Sir is presently working as a principal scientist and associate professor in the Department of Molecular Microbiology and Immunology at CSIR Central Drug Research Institute, Lucknow. His research focuses on complementary issues of drug discovery for MDR bacterial pathogens and delineating antimicrobial resistance mechanisms prevalent under Indian conditions. His lab specializes in screening of diverse natural products as well as synthetic organic and inorganic compounds for their antibacterial efficacy from in vitro to animal models of bacterial infection. To better understand the existing and prevalent resistance mechanisms, his lab extensively characterizes the clinical isolate at both the genetic and phenotypic levels. Together, this data can help guide a more defined approach for successfully identifying the molecules with potent activity against MDR pathogens. So, with this brief introduction, I now request Dr. Sidharth Chopra sir to begin with his talk.
Hello. Am I
Yes,
audible?
sir. Yes, sir. You are audible.
Okay, great. Thank you for the very nice introduction, Dimple. And I really appreciate your kind invitation to present at this two-day lectures, series. I would now like to share my screen. Mm, mm, are my slides visible?
Yes, sir. The screen is visible,
Okay.
and even the first title slide is visible.
Slightly visible. Thank you so much. If you guys don't mind, I'll turn my camera off. It's raining here quite heavily, so our network is extremely poor. So it's possible I might get logged out in the middle. So I'll turn my camera off. So thank you very much for the very kind invitation. I am now going to, so the title of my talk, is Drug Repurposing, a New Front in the War Against Bad Bugs. And the reason I have put this title, and I'm really happy that the audience participated very nicely in Dr. Karishma's talk and asked very pertinent questions. And so one of the things which she very well represented in her talk was the fact that it seems like we are in a war with bacteria, which is a continuous war. And because of the small generation time as compared to ours, at present, it seems like the bacteria are winning. And so the only viable answer to to antimicrobial resistance is the continuous discovery and the development of new drugs. Because so as I will explain in my slides, these are the thing. So AMR has been defined by WHO and multiple international agencies as one of the biggest threats to global health, food security, and the development of mankind. This is not a trivial statement which is made by the WHO. It is one of the biggest problems. So if we think from a slight history, the first antimicrobial which was discovered was discovered by Paul Ehrlich, and he used it for the treatment of Treponema pallidum, which causes syphilis. On the other hand, the first commercialized antibiotic was discovered by Sir Alexander Fleming, and as you might all know, it was a fortuitous, the discovery, because he had gone on a vacation, came back, saw a mold which was inhibiting a culture of Staph aureus. From that extract, he was able to isolate the natural product, which he named penicillin, which was then commercialized right after or right during World War II, and helped to save a lot of people's lives, especially injured soldiers. So that is why.
Sir, your voice is breaking in between. Your voice is breaking in between.
Okay. Do you want me to log in and log back again? Is my voice ahead better now?
Yes, sir.
Okay, great. Thank you. So as I was explaining, the first antibiotic which was used in the clinic was discovered by Paul Ehrlich, whereas the first commercialized antibiotic which was discovered was by Alexander Fleming. So I'm going to go into a little bit of basics. What are antibiotics? Antibiotics are basically medications which are used to treat bacterial, viral, or parasitic infections. And the way they work is by having selective toxicity, in the sense that a drug that kills harmful microbes without damaging the host. Therefore, as one of the questions which was asked by the audience, at the start of the antibiotic the therapy, the infection should be cultured to identify the causative organisms, and a potential antibiotic susceptibility test should be conducted. Without that, we are basically misusing the power of antibiotics. And as I'm very sure you guys all know, there are three methods to control the spread of infectious disease. The three methods are: one is drugs, the other is vaccines, and the third is diagnostics. Drugs, we talk about a lot. We talk about antimicrobial resistance. Vaccines, we have seen the power, for example, with COVID. But the least thing which is talked about are the diagnostics. And that, unfortunately, is the Achilles heel of why we are seeing greater and greater amounts of antimicrobial resistance. So what is the difference between susceptibility and or resistance? Susceptibility is the ability to treat a specific bacterial infection with an antibiotic, and it often has a high probability of a favorable outcome. On the other hand, resistance is the inability to treat a specific bacterial infection with an antibiotic and is typically associated with a very poor outcome. If you see the picture below, this is a very simple zone of inhibition assay where once the bacteria are cultured, it shows you a zone of clearance that indicates a susceptible antibiotic, whereas if there is no zone of inhibition, it indicates resistance. So, is antimicrobial resistance a major problem? Is it a global problem? What are the costs associated with it? This question has been asked since the last 30 to 40 years. And in order to actually put a financial cost to this and to determine the scope of the problem, the UK government in 2014 to 15, they released a report by Lord Jim O'Neill, where they did the prediction that by 2050.
Excuse me, sir.
Yeah.
Sir, actually, your slides are not visible.
Okay.
Your screen is not visible.
Okay. Let me do it again, please.
Yes.
Are they visible now?
Not yet, sir.
Okay.
We can see just the black screen.
Just the black screen? Okay.
Yes.
Let me share
Yes.
again. There seems to be an internet issue here, because I'm able to see everyone, but my slides are not getting visible. That is very strange, but okay.
Sir, if it is okay with you, then you can share the presentation to Ekta ma'am. She will present the screen for you.
What about now?
Yes, yes. Now it is visible.
Okay,
The
great.
antimicrobial resistance problem slide.
Exactly. Great.
Yes,
Awesome.
it is visible, sir. So
And
you may continue now.
Yeah, I will continue now. It is visible to you guys?
No, sir.
Yeah, that's what I thought.
Sir, will you please mail your presentation to Ekta ma'am? She will present the screen for you.
Yeah, I will do. I will do it right now.
Yes, because I guess you are facing a lot of connectivity issue.
Yeah. I have mailed it to Ekta.
Yes, sir. So ma'am will present the screen.
Okay.
So we have to just wait for a while.
Absolutely no issues. I'm going to try one last time if it works. Otherwise, you guys just do the present the slides, and I'll just speak from here. Are they are they visible now?
Yes, sir.
Yes, sir. Now the slide is visible actually, but
Okay.
it may
It may
again.
break down again. I
Yeah.
know. I'm I'm sorry. I don't know why it's happening. It works out easier than this. I don't know if it's an internet issue or some other issue is there. I guess
Sir, but now the slides are properly visible. Means
they
we
are
can see
visible.
the slide.
They are
Yes.
okay. So I'll
Yes.
start. I'll do the presentation now.
Yes.
So we are at the antimicrobial resistance. You know the slide, right? Where it shows the world map.
Yes, sir. Yes.
Okay, great. Okay,
Yes, sir.
okay, great. So uh, in the 2015 report by Lord Jim O'Neill, they postulated, they not hypothesized, they predicted that by the year 2050, there will be almost close to a massive number of cases in the whole of Asia. As you can have a look, almost there will be close to 4.7 million infections, which are visible here. And unfortunately, Asia and Africa will bear the brunt of the maximum number of drug-resistant infections. Even bigger problem, the cost to the world economy will be something close to 10 to the power $14, which is $100 trillion, which is a huge number. Because as you can see with the COVID, there will be lockdowns, there will be high mortalities, and please remember, unlike COVID, where there is no drug resistance as a factor. If, for example, there is a drug resistant pandemic, the cost in terms of morbidity, mortality, damage to the economies, global recession will be much higher, much higher. And so, but unfortunately, unlike COVID, which is a straightforward microbiological problem, antimicrobial resistance is not that straightforward, because antibiotics are utilized in animals, in agriculture, in aquaculture, for veterinary use. They are used in medicine with prescription, without the prescription. And the biggest problem in this is the over-the-counter sales. They are present. They are A lot of the antibiotic kind of residue is present in the wildlife, is present in water, it's present in the soil. People who choose to travel for medical reasons, for example, medical, the tourism, they bring the bacteria, viruses, fungi with them, and they take it with them as well when they go back. So it allows it to spread. And like the question was being asked, why new antibiotics are not entering the market is because primarily because of lack of profit in this the sector, in this domain. The science is extremely challenging and difficult, while the regulations are getting complexer and complexer by the day. All these factors combined make AMR an extremely challenging multi-stakeholder problem. And as we have seen, the world doesn't do kindly to multi-stakeholder problems. So what does resistance look like? So antibiotics typically target lots and lots of targets within the bacterial, the cell, but there are 5 major pathways which are targeted. The first one is the cell wall, which is targeted by beta-lactams and vancomycin. The other is DNA RNA synthesis, which is targeted by the fluoroquinolones and the rifamycins. The folate synthesis is targeted by trimethoprim and the sulfonamides. The cell membrane is targeted by antibiotics by lipopeptides like daptomycin, and the protein synthesis is targeted by a number of antibiotics, including linezolid, the tetracyclines, macrolides, and aminoglycosides. And because these are the 5 most common targeted pathways, the resistance mechanisms are also quite complex, and they have existed since time immemorial. The first one is
Sir, the slide is not visible again.
Okay. Ekta, have you gotten the slide?
Yes, sir. I'll share the screen then.
Please, thank you so much.
Yes.
It will be a PDF. Just go to the view and just go to the view. I will open it here. Just go to view in the full screen mode.
Yes, sir. Now you may continue.
Okay, thank you so much. So we are on the slide which says, what does the resistance look like?
Yes, sir.
Great, thank you.
Exactly that slide.
Exactly that slide. Thank you so much. So I'll say just move to the next slide when I'm done with this, okay?
Yes, sir.
Thank you. So what are the various targets which are targeted by the antibiotics, as well as what does resistance look like? So there are 5 most common targets which are targeted by the bacterial pathogens. In any bacterial pathogen, they include the cell wall or the peptidoglycan. They are targeted by beta-lactams and vancomycin. The DNA or RNA synthesis is targeted by fluoroquinolones or rifamycins. The folate synthase is targeted by trimethoprim and sulfonamide. The cell membrane is targeted by lipopeptides like daptomycin and polymyxin B, whereas the protein synthesis is targeted by several antibiotics like linezolid, tetracycline, macrolides, and aminoglycosides. Because there are these many targets, obviously the resistance mechanisms also are quite explicit and include efflux, that is the most common one. Their targets are fluoroquinolones, aminoglycosides, tetracyclines, beta-lactams, and macrolides. Lack of impermeability or change in the cell wall and membrane that impacts the tetracyclines, the trimethoprins, vancomycin. Modification of the target impacts a wide range, as well as the release of inactivating enzymes like acetyltransferases or methylases impacts the beta-lactams, aminoglycosides, macrolides, as well as the rifampicins. Next slide, please. So what are the various mechanisms of AMR? So AMR can be intrinsic or it could be acquired. And if it's intrinsic, that basically means the organism's genome automatically includes the resistance mechanisms, whereas the acquired the resistance when acquired has two ways. Either there is mutation in the chromosome, or there is the presence of extra chromosomal plasmids. Next slide, please. So intrinsic resistance is the one which occurs naturally. For example, one mechanism is lack target. For example, mycoplasmas. Mycoplasmas don't have the cell wall. That's why they are innately resistant to penicillin. The other major mechanism is the presence of innate efflux pumps. The drugs are blocked from entering the cell, or increased export of drug so that it does not achieve adequate internal concentration. For example, E. coli and Pseudomonas are the most common pathogens which have these efflux pumps. And the third is the release of the enzymes which inactivate the drugs, for example, beta-lactams or the cephalosporinase in Klebsiella. Next slide, please. On the other hand, the acquired resistance is mediated by either mutations, which refers to the change in DNA structure. It occurs at a frequency of 1 in 10 million cells. And the major examples are, for example, MTB, Mycobacterium, the leprae, MRSA, which is methicillin-resistant Staph aureus. Often the mutants have reduced susceptibility to a particular antibiotic or an antibiotic class, for example, the fluoroquinolones. The other way to acquire the resistance are plasmids. Plasmids are defined as extra chromosomal genetic elements that can replicate independently and freely in the cytoplasm. These are often called as the R genes or the R plasmids, and can be easily transferred from one organism to another organism. And the advantage is they can either integrate into the genome, so they are stably transported into daughter cells, or can replicate as independent plasmids. And much of the drug resistance encountered in the clinical practice is often plasmid mediated. Next slide, please. The mechanisms of resistance gene transfer. So the R plasmids or R genes can be transferred from one bacteria to another through 5 different mechanisms: conjugation, transduction, transformation, and in addition to plasmids, there can be transfer of genetic material by transposons as well as by integrons. So the third mechanism is the lack of the permeability of antibiotics. For example, majority of the antibiotics enter the bacterial cells via the porine channels in the cell wall. So the organism loses these porine channels. There is a cost to pay. It grows a bit slower, but now the antibiotics are not able to enter the cell. The efflux pumps are one of the major mechanisms. They include a huge variety of efflux pumps, including the ABC superfamily, the MFS, the MATE family, the R&D family, the ABC super family, and these are one of the most well-studied mechanisms where the antibiotic concentration inside the cell is not allowed to build up to actually inhibit the growth of the cells. And there is literally no family class which is not affected by these efflux pumps. Next slide, please. The fifth mechanism is there is a structural modification of the antibiotic target sites. What happens is the bacteria change the targets by either mutations or removal or by deletion, so that antibiotic target is absent. So that, for example, like in mycoplasma, there is no cell wall, so penicillins, they cannot work. Next slide, please. So, and the next mechanism is modification or protection of the target site. The target sites most commonly are the ribosomal point mutations. The antibiotics which are affected are the tetracyclines, macrolides, and the clindamycins. DNA gyrase, when mutated, the organism becomes resistant to fluoroquinolones. The penicillin binding proteins, which are required for the synthesis of the cell wall, when they are modified, there is resistance to penicillins. And when the DNA dependent RNA polymerase is modified, there is resistance to rifampicin. This is irrespective of the bacterial class and occurs in all bacterial species. The last but not the least is the major enzymes which are produced by various pathogens which inactivate the antibiotic, for example, either by acetylating it, methylating it, or simply by degrading it. So these enzymes are often released into the extracellular medium where they damage the antibiotic before it has a chance to enter the cell and damage, cause either the death or stasis of the bacterial infection. These examples most commonly are like the aminoglycosides, where there's modification by the phosphorylation, adenylation, or acetylation. The erythromycin and the tetracyclines, there is a change in receptor by methylation of rRNA, or there is a reduced uptake or increased export. The sulfonamides are also impacted because there is an increased export out of the cell combined with reduced affinity of the enzyme. So how bad is AMR? We know that there is a lot of AMR in the world. How bad is it in the Indian scenario? So to understand that, what we have to keep in mind is the fact that it takes almost 20 years of research and clinical work to get a drug into the market. The cost is around $1.2 to $1.5 billion, whichever company puts in that much amount of the money wants to recoup that. So, but unfortunately, because of the indiscriminate utilization and really high rates of AMR, in many cases, AMR appears in less than 6 months. Thus, there is not enough time for the companies to recoup their investment in this area. That is one of the biggest reasons why major pharmaceutical companies are exiting the infectious disease area, right, left, and center. And as was the question was asked in the previous talk, there used to be a total of around 20-25 companies heavily invested in antibiotics, but now the number has come down to 2 or 3, and even they are considering withdrawing. Whereas the reality in the clinic is the mortality rate from infections is rising unceasingly, and it is putting a lot of the pressure on the various hospitals, which increases the cost. And for example, like Dr. Karishma's lab, which works on the Acinetobacter baumannii, there is also almost 100% increase, which makes it the double the cost of treating drug-resistant Acinetobacter infection, whereas there is almost a 20 to 25% more chance of mortality with Acinetobacter, which is resistant to multiple drugs. Next slide, please. And I have taken this from the prediction that there will be almost 10 million more deaths, and that is, believe me, a very conservative, the prediction, because as we see in almost all the big tertiary care hospitals these days, majority of the inpatients and even outpatients are getting lots and lots of drug-resistant infections for which the last line of drugs are getting exhausted. And to counter these things, WHO has released a list of approved pathogens in 2018, which are basically most of the escape family, and the best part is that there are very, very few drugs in clinical trials against these pathogens. This chart on the right, it shows you the drugs when they were approved for gram-positive pathogens. And the bigger worry is that the gram-negative pathogens, which is a part of the um, uh, escape pathogen panel, there are 4 gram negatives and 2 gram positives. And majority of the antibiotics in clinical trials or which are entering the market target gram positives. There is simply no efficacy against gram negative pathogens. Next slide, please. On the other hand, the antibiotic utilization in India is continuously increasing day in, day out, primarily because of the fact that we live in very unhygienic conditions, and there is a lot of unregulated over-the-counter sale as well as over-the-counter utilization of late-stage antibiotics like carbapenems and the cephalosporins without much diagnostic backup. And this trend is reflected in the charts which are shown here. As you can see, in India, almost 75% of the Acinetobacter species are resistant to carbapenems, which are the last line of defense. And the only treatment option which is available for these pathogens then is polymyxin B, which is neuro and nephrotoxic, thus damaging the nerves and the inner ear canal of the patients. And as you can now see, even Klebsiella, these charts are a bit older, but even now, the latest reports from ICMR indicate that almost 80% of all the Klebsiella isolates from hospitals are heavily resistant to even polymyxin B. And with the discovery of the MCR1 plasmid, which transmits the polymyxin resistance on a plasmid instead of it being genetic, has really worsened the situation. Next slide, please. The biggest point in this is that in 2010, India was the largest consumer of antibiotics, where we consumed almost 3 logs more antibiotics than people in either Europe or USA, which continuously puts the indescribable kind of the pressure on the bacterial pathogens and not only I keep talking about bacteria, but the same situation, actually worse situation, exists for the fungi and viral infection. And specifically with the COVID, we have seen there is a lot of illogical utilization of hydroxychloroquine, ivermectin, all these drugs which are extremely important for the treatment of different infectious diseases like malaria, parasitic worms, are being indiscriminately utilized in the hope that they have an effect on the virus, where there is no scientific rationale behind their utilization. And in this situation is not only in the Indian subcontinent, it's also there in the BRICS countries, which are supposed to be the next big development countries. 23% of the increase in retail antibiotics was observed, out of which 57% increase was in the hospital sector in China. So these numbers are extremely, extremely damaging and serious. Next slide, please. And for example, our doses went up between 2001 and 2010, went up by 62%. That's a very high number. We were at 2010 consumption, and these are just talking about pills. They're not talking about injectables at all. So 12.9 billion as compared to China, which is twice our population, 10 billion. US, which is 1/3 our population, 6.8 billion. These are huge numbers. You cannot imagine the pressure on the bugs to mutate the last line of drugs. And the worst part is that majority of the people are not clear that antibiotics do not work on viral pathogens. They should take the complete course of antibiotics. That also is not clear to majority people. They just take one dose, they feel better, and they move on. And that is one of the biggest issues. Patient non-compliance is one of the biggest issues driving AMR in the world. Next slide, please. So AMR has been covered in the clinic, by the clinic, in the newspapers, in the scientific, over and over again. Next slide, please. And just to come to specifics, the mortality odds among patients with resistant bacterial infection are extremely high when it comes to uh, patients which are infected with either MDR or XDR bacterial isolates. In a survey done in India during 2007 to 2011, almost 98% of the isolates were resistant in Lucknow in 3,000 blood samples to ampicillin, which is one of the primary lines of defense. Almost 92% were resistant to penicillin and erythromycin in Bangalore. In Delhi, there were 10 to 20% which were resistant among 83 OPD cases. All these numbers basically tell you that there is a lot of indiscriminate utilization of antibiotics. The diagnostics are extremely lacking, and our entire approach for the treatment of infectious disease relies on the continued working of antibiotics. We do not have an alternative, and we will be paying very dearly for this lack of alternatives very soon. Next slide, please. And let's not get into the discussion about the difference between the health care associated infections as well as the community associated infections. For example, the accepted rate of healthcare infections, which basically means the patients when they are admitted to hospitals, the rate is around 10% of them will get infected. In India, it has been estimated that almost 20%, and that's a number which has come in the newspaper, which tells you automatically that it is massively underrepresented. They get infections. And in premier institutes like AIIMS, New Delhi, in SGPCI, the number is 20%. In government hospitals of UP, the presence of hospital-acquired infections is 40% or more. And in every 100 cases of that, the majority are UTIs because of the catheters, 25% are the surgical site infections, and the majority are the ventilator-associated pneumonia due to lack of hospital stewardship, the pressure of the patients on the hospital, and as well as the fact that there is a lot of bacterial pathogens in the clinic which are resistant to various treatments for their killing. Next slide, please. And AMR is just not that there is no antimicrobial. AMR is because there is too much antimicrobial in the environment. That actually comes under the One Health approach. For example, that next slide, please. So, is resistance new? We keep talking that AMR exists. Okay, we acknowledge that AMR exists, but now my next question is AMR new? No, the answer is very clear. Penicillin was discovered in the 1920s. The first resistant isolate was identified in 1941. And that is when it was commercialized only in 1938. So 3 years after clinical utilization, right after during World War II, 1941, the first resistance was isolated. Lipopeptide that's the last antibiotic isolated in 1987, 2005. The first resistance was there. So as you can see, with lots and lots of antibiotics, they are there. So there is no antibiotic for which there is no resistance. There can be slow resistance, there can be fast resistance, but the bugs will find a way. And the reason why the bugs, they find a way, next slide please. So is the fact that, so we have what is called as a National Action Plan on AMR in India, which was released by the Ministry of Family and Health and Family Welfare by Government of India. And it tells you that there is a lot of resistance. You have to be careful. You have to not take the over-the-counter drugs. You have to go to a doctor. The doctor has to do proper diagnostics, and then they have to prescribe you the antibiotics. Next slide, please. Unfortunately, there is very little money in the National Action Plan on discovering new antibiotics. Next slide, please. So in a call to that, there were two, the declarations. One is known as the Jaipur Hedge Declaration, which said that if there is increasing AMR, which will lead to lot of bacterial infections, including drug-resistant infections, India will not be able to achieve the minimum Millennium Developmental Goals, specifically that address containment of HIV/AIDS, TB, malaria, and other bacterial pathogens. That is why there was the commitment from the government saying that we need to fight against antimicrobial resistance. However, again, as usual, it was just words. There was no money spent to actually do that. As a result, there were increasing number of deaths in cancer patients, which are considered to be immunocompromised. And that is when a bunch of doctors who actually treat cancer patients day in, day out, they realize that they have to get it to the ICMR's attention that the lack of new antibiotics is really harming. And that is when the Chennai, the declaration was made by Dr. Abdul Ghafoor, who's the coordinator for this Hedge Declaration, and it called upon the Ministry of Health and Welfare to actually encourage the Indian industry to identify new antibiotics. Next slide, please. So if we look at the numbers, so almost 30 million pounds of antibiotics are utilized in animals, whereas only 7 million pounds are utilized per year in humans. So that automatically tells you that even if we are able to find new antibiotics and commercialize them, the pressure on the bugs will remain irrespective of the fact, because 3 times or 4 times greater are utilized in animal feed. That is why the government of India has recently banned the utilization of colistin and other antibiotics which are used for treatment of infections in humans, in animals. But the reality is that these names are explicitly not mentioned on the food, the packaging, which is used for the growth of various farmed animals, and often there is indiscriminate utilization. So the idea behind this is that the fact that the gene travels from the gene is able to travel from the animal to the farm sector. We eat the animal, it comes into humans, it goes into the potatoes, tomatoes, and the onions, and then we end up consuming the antibiotic residue as well as the antibiotic resistant gene, which leads to increased resistant infections in humans. And that is why there is an increased reliance on the One Health approach to study AMR. Next slide, please. So is AMR new? There was this very beautiful study done by the group in Canada, which answered this question extremely nicely and said the fact that there is lot of resistance was there even during earlier times. What they have done is that they went to Alaska into the permafrost and they captured the ice cores, and the ice cores, as you can see, contained the DNA for all sorts of old animals which don't exist anymore, and along with the animals it consisted of the antibiotic resistance markers. Next slide, please. And when these old antibiotic markers were analyzed by genome sequencing, it was found to be that they contain the huge resistance to antibiotics which are being used in the clinic right now. Next slide, please. And as an example of that, this group led by Dr. Costa expressed these vancomycin resistance elements in the current human pathogens and expressed resistance to antibiotics. This work is remarkable for this reason, that when these resistance elements were found There was no concept of vancomycin as an antibiotic. There was no concept of vancomycin to be used in the clinic because there was no concept of the clinic, and humans did not exist at that point of time. So that tells us everything we need to know about AMR being a natural phenomenon which has always existed since time immemorial, because if we remember, majority of the antibiotics are natural products made by one bacteria to fight another bacteria for the limited food available in the environment. Without the environment, without these antibiotics, there would be no food left. And that is why what we have done is we have started using their chemical messengers as antibiotics to get rid of the infection. And that is why there is a much increased rate of generation of resistance. Next slide, please. So how to fight AMR? Policy, as I have told, there is multiple AMR, the declarations, there's a policy. It is completely useless until it's implemented, that too with an iron rod in a country like India. The mentality has to change. Drug discovery is an important facet of science. If not now, then when? Do we really want to be at a stage like we are currently with COVID, where we do not have any drugs to fight it? Because COVID is not that damaging when it comes to mortality. Imagine having a gram-negative bacterial infection which damages your kidneys, and then you are really screwed. So drug discovery has to be practiced earnestly. Sanitation, Swachh Bharat is the key. It has been the key all along. If we continue to live in unclean environment, if we continue to live the way we are living right now, this will not be easy to pursue. And in the end, healthcare decisions should be based on humanity, not economics, because economics is mathematics, and mathematics is not the right way to decide who lives and who dies. So in answer to the audience question, in the 1970s, there were these many companies working to discover new drugs. In 1990s, the field had thinned out. In 2010, only these 9 companies were making antibiotics. And in 2020, only 2 are left. One is Merck, the other is Pfizer. Nobody else has an active antibiotic discovery program. Majority of the discovery is being conducted by small biotech-based companies who do not have any product in the market other than the one they are working on. And so they are totally dependent on venture capital money, which is unstable at the best of times. That is why there has been increasing news in the recent years, in the last couple of years, that even companies which have very potent drugs in the clinical trials are going bankrupt, because no investor is interested in putting the money there, because they see stocks like IT stocks where you can make an app and earn a million in a week. Whereas if you put the money in drugs, it takes 10 to 15 years for them to recover their money. Next slide, please. So now I'm going to give you a little bit of a background on how antibacterial drug discovery is conventionally done. Next slide, please. So drug discovery is a long work. It starts with screening. There's a lot of interdisciplinary work. There's a lot of screening, microbiology, chemistry. Once the drug is identified, it goes into the preclinical phase and followed by the clinical trials, followed by FDA review. So on an average, if you start with 10,000 compounds, you will get 1 FDA approved drugs. On an average, 15 years. On an average, around $500 million to $2 billion. Next slide, please. So the way to generate the key starting material for any antibiotic drug discovery program or anti-cancer or any program is the quality of the chemical libraries. That is simply the biggest challenge facing all the companies in the world. On top of that, because the amount of the chemistry which can be done in the lab is limited, and it has to be economically viable, it has to be transferable, it has to be industrially scalable. that rules out majority of the natural products because they are extremely complex to synthesize, and often we cannot do the synthesis. So we are dependent on the microbes which make them. So there are two ways to screen. There is one which is called as a target-based screening, where you know the target. For example, you want to target the DNA gyrase, then you use bioinformatics and you dock the various structures in there in the aim of identifying something. The other is unbiased screening, which has been followed from day one, which is the whole cell screening. The advantage of whole cell screening over the target-based screening is the fact that in whole cell, if you get growth inhibition, then you know that the compound under question has the ability to cross the bacterial wall in the membrane. It actually interacts with a specific target and is able to kill the cell. The drawback is you don't know what it's hitting. In the target-based screening, the advantage is you know what it's hitting, but then you have no clue whether it will be able to enter the cell. Will it be able to interact with that target when it is present inside a whole bacterial cell, and whether it will lead to any biological effect. Next slide, please. Or we can have unconventional the discovery, which was also told by Dr. Karishma, that there are natural head products, there are ethnobotanical knowledge, there are traditional medicinal plants, there are endophytes, polyketides, small peptides. Next slide, please. Unconventional discovery typically relies on a lot of combination of fields, including bioinformatics, metabolic engineering, different carbon sources, often leading to fermentation, which leads to identification of a natural product or a new species, which can then be hopefully converted into a new antibiotic. On the other hand, there is another mechanism where you, instead of dealing with a single organism, you deal with microbial communities, you do co-cultivation, you do a lot of mass spec, a lot of LCMS, identify the structures which were told by Vinogradsky column. He was a Russian microbiologist who said that you have to co-cultivate organisms, otherwise they will not make these secondary messengers. Next slide, please. One of the most beautiful example of unconventional, the discovery is the discovery of the drug called as the Texicobacter. The molecule called as Texicobacter, which was discovered when the scientists working in Boston, USA, were able to co-culture multiple organisms in a single chip, like the one shown above, which is a black enclosed chip. And the advantage was that there were multiple microbial communities cultivating in there, and this molecule was released into the environment as a result of the competition between the co-cultures. And this antibiotic kills pathogens without detectable resistance because it targets an essential part of the cell wall synthesis pathway. Next slide, please. The next approach, which is typically utilized with existing antibiotics, is the drug combination. So the advantage of the drug combination is that there could be synergism, specifically like the beta-lactams and the aminoglycosides. One targets the cell wall, the other targets the protein synthesis pathway, and there are disadvantages as well. For example, tetracyclines and the penicillins often induce bacteriostatic effect. Remember one very important thing: if the drug combination, or even if a drug is static, it just indicates that the bacteria have more time to mutate and generate resistance. Next slide, please. The next biggest alternative to antibiotics, I don't want to say alternative, I want to say another way by which we can target pathogens, is the discovery of antimicrobial peptides. These are natural amino acid or unnatural amino acid containing peptides which target typically the membrane or a specific target within the cell. There are large number of examples like lysozyme, brevin, protegrin, dorsomycin, defensins, and the biggest problem with these is that it's extremely challenging to scale these up in enough quantity to conduct unbiased clinical trials, and most often than not, these work only as topical formulations. Next slide, please. This list gives you an example of all the various peptide formulations which are right now in various, the clinical trials, phase 1 and phase 2. And as you can see, they are all for really topical application. There are extremely very few which can be utilized for intravenous or parenteral applications. Next slide, please. So, sir, there are some newer antibiotics in the market, like linezolid was commercialized in the year 2000. Tigecycline, both of them inhibit the protein synthesis. Daptomycin, dalbavancin, the telavancin, ceftibiprole, and the ceftaroline, as well as the ecloprim, they all inhibit either the cell wall or the DHFR. Next slide, please. One very important aspect of new drug discovery is the fact that we have lots and lots of drugs for which antibacterial efficacy has not been found. This comes under drug repurposing, because the advantage is we can use the knowledge already the FDA has for drug repurposing. Next slide, please. What is repurposing? So repurposing is the process of application of an existing therapeutic to a new drug indication, and it's also called as drug repositioning. The advantage is that in the de novo drug discovery, which takes the 15 years, the drug repurposing can take around 2 to 4 years, because there is no safety concern. There is very less chance of failure in the clinic, because already the pharmacokinetics, pharmacodynamics, and all the clinical the parameters have already been done, and that leads to a reduction in the cost and time, which increases the chances of it being approved. Next slide, please. What are some of the examples? Some of the most potent examples, not only as antibiotics. For example, zidovudine was originally indicated for cancer, but now we know that it is a very effective HIV/AIDS drug. For example, the sildenafil, which was originally indicated for angina, is now used for the MED, which is male erectile dysfunction. The other drug which is used is methylamine, which was used originally for severe hypertension, is now used for the treatment of attention deficit hyperactivity disorder. Next slide, please. So this slide also gives you. So next slide, please. What is the need for repurposing? The advantage is that drug development can be time consuming and expensive. It takes a lot of time. Drug repurposing, because of the fact that a lot of the work which needs to be done for a new molecule has already been done. Therefore, existing drug with a new indication is always a better option. Next slide, please. What are the advantages of drug repurposing? The advantage are that there is very fast bioinformatics, biology, big data, which can be conducted, saves time, saves the years. That is why it's a better option. Next slide, please. Next slide, please. Next slide, please. Next slide, please. I'm going to give you certain examples of drug repurposing, which we have conducted in our lab. For example, this drug, which is called as ivacaftor, we collaborated with Dr. D.S. Reddy in NCL in Pune, and this drug, ivacaftor, is utilized for the treatment of a very severe lung disease in patients, and at that point of time in 2017 was one of the most expensive drugs on the market. We found out that it has very potent activity against Staph aureus, which is an escape pathogen. It's a priority 2 pathogen with that, but it is only active against gram-positives. The best part is it works irrespective of the fact of the drug resistance. As you can see in the table, it exhibits concentration-dependent bactericidal activity. Next slide, please. And when we tested it against bacteria which are in the biofilm or which are in the macrophages, it has very good efficacy, which is comparable to vancomycin, which is standard of care. And when we tested it in animals, there is in the in vivo murine neutropenic thigh infection model at an equivalent dose as compared to vancomycin, there is a better reduction in the colony counts. This is one example. Next slide, please. We repurposed another drug, which is diphenylene iodonium chloride, or DPIC, against Staph aureus. So DPIC is a nitric oxide synthase inhibitor, which is not a drug at all. It is a molecule which is used in the lab. We repurposed it for its antibiotic activity. The advantage is it works beautifully against MRSA versa strains, which are resistant to lot number of drugs. It exhibits concentration dependent bactericidal activity. Next slide, please. And when we tested it against mycobacterial pathogens, it is extremely potent, as you can see in, for example, against H37RV, which causes tuberculosis. The MIC is 0.03, which is comparable to INH rifampicin, which are the frontline drugs. We tested this molecule in the in vivo activity in murine neutropenic thigh infection model, and at 25 times less dose than vancomycin, it is able to cause similar reduction in the activity. This we published long ago, and now we are in the stage where we are collaborating with various medicinal and chemists, and have identified newer analogs of this molecule, which are even more potent against gram-negative infections. Next slide, please. So these are two examples. We have also repurposed lots of other drugs like disulfiram, which are used for the treatment of alcoholism as very potent Staph aureus inhibitors. We have repurposed one additional drug as a very gram-negative infection. And all this to tell you that discovery of a new molecule is extremely challenging. If we can identify and repurpose already existing drugs against drug-resistant bacterial pathogens, it might give us the time to discover new drugs. So, for example, in the treatment of drug-susceptible TB, there is INH, ethambutol, pyrazinamide, and rifampicin. But on the other hand, when the case becomes MDR, it becomes resistant to INH and rifampicin. However, there are still thiocyanamides, fluoroquinolones, and cyclic peptides which are utilized. Next slide, please. When we go to XDR TB, there is literally nothing which works. And that is a very worrisome situation, because majority of the MDR patients which are coming back to the clinic have XDR TB. Therefore, they are being given extremely toxic drugs in the hope that they will get a better efficacy. Next line, please. This is a very powerful reminder of the way TB used to be treated, which was the utilization of the sanatoriums. We are seeing very similar treatments being afforded for COVID. And if we want to really fight AMR and not come back to the pre-antibiotic era like this, drug discovery, drug repurposing, peptides, phages, everything has to be used in conjunction, and everything has to be used wisely, not indiscriminately. Otherwise, we will be back in this situation where even a very simple cut can be life-threatening. Next slide, please. And the last slide. This is a quote from a very wise man in his 1945 Nobel Prize lecture. Fleming himself had warned of the danger of AMR. He said, it is not difficult to make microbes resistant to penicillin in the lab by exposing them to concentrations not sufficient to kill them, and the same thing has occasionally happened in the body. And the person who does this makes his microbes drug resistant. Thank you. I am now open to taking questions.
Thank you so much, sir, for conducting such an informative session. I'm sure that all the students are truly benefited by today's session, and I hope that the mechanism of antibiotic resistance and the knowledge about the drug repurposing, as well as how to overcome the challenge of antibiotic resistance, is clear to all the participants.
Thank
But
you.
still, if you have any doubt, students, you may ask the doubt by unmuting yourself, or else you can also post the message in the chat box. So we will move to the question answer session now. So students, you may ask doubts now.
Excuse me.
Yes, Mohit.
Yes, ma'am. I would like to ask a question to the sir. Thank you, sir, for such an interesting session. Sir, my question is from your talk only. Sir, somewhere you quoted that antibiotic resistance and the direct and indirect effect will be more on the Indian and African community. So
Yes.
my question was that, sir, we also know that many of the concurrent reasons is making the human colonies inhabiting the African continent and Indian subcontinent are very resistant with respect to individuals which are living in developed nations like America. So, sir, is there any scope for evolution of antibiotic resistance or any sort of resistance in the microbes because of the immunity of human itself? So, my background of this question was that basically some of the very contagious viruses like Ebola virus and even recent Omicron has been came from the African soil. So, sir, is there any scope of human immunity playing in a very essential role for developing such resistance in microbes? Thank you, sir.
Thank you, Mohit. Very nice question. Very interesting question. It has a lot of implications. Unfortunately, when it comes to a direct competition, one is to one competition, um, microbes and humans cannot compete on the same time scale. One organism reproduces in 20 minutes, the other takes 30 years. So these time scales are not compatible. Thus, human immunity can play a role, yes, but it will be very slow. And to the fact you asked that Omicron has come from the African, the soil. Um, I would like to say that, to be honest with you, we had lucky that the South African scientists were able to catch Omicron. Um, I'm very sure that if it had to come up in any other part of the world, it would not have caught so easily. So I don't think they should be penalized for what they have done. They have done pure science and tried to help everyone. But so I don't think human immunity can catch up in a timely manner.
Yes, sir. Thank you.
Any more questions?
Sir, I have a doubt.
Please.
Sir, we talk about the antimicrobial resistance in pathogen.
Uh-huh.
But sir, is there any scope of research by using the ancient techniques by chanting mantras or something so that we can overcome the problem?
Very nice question. I think the mantras can probably help you achieve a calmer state, which can possibly have an effect, but I don't think it can be utilized in the clinic.
Okay, sir. It was out of curiosity. Thank you, sir.
Not a problem. Any more questions?
Yes, students, do you have any questions? Any other question? You can post in the chat box. I guess there are no more questions. So once again, thank you so much, sir, for this session. And now, yes, and now I request the participants to please turn on your cameras for a group photo. Everyone, please turn on your cameras. Okay, and the link for the feedback form is already posted in the chat box. So you can fill this feedback forms, and now the session is going to continue tomorrow also. Okay, so stay tuned. Tomorrow we are going to meet two new speakers. So for now, we'll stop over here, and I declare that today's session is ended. Thank you so much, sir.
Thank you very much. Thank you very much.